Welcome back, students, to our continued lectures on cloning vectors. We'll be discussing first lambda as cloning vectors, cosmates, high capacity cloning vectors like back, and yak, expression vectors, type of promoters we use for cloning, and use of linkers and adapters during cloning. We have already understood the gene organization in the first lambda genome, how genes regulate expression of lytic and lysogenic cycle. There are genes present in the middle part of the genome of first lambda which can be removed and which can be manipulated, and foreign DNA can be expressed in that part. The dark part here shows the region of fast lambda which can be excised and can be replaced with foreign. Two types of vectors are possible: insertional vectors and replacement vectors. In case of your insertional vectors, you can see there is one restriction sites which can be digested and where the foreign DNA can be inserted without removing any part of the lambda genome. On the other hand, in case of replacement vector, you can see two restriction sites. By digesting the vector with restriction sites, restriction enzymes, you can remove that dark part and which can be replaced by your foreign DNA in case of your replacement vectors. Wild type lambda DNA contains a eco R1 site at the CI gene which can be digested and which can be used as cloning site for as insertional vectors, which is shown here. In case of your substitution vector, the stuffer fragment can be removed, polylinkers can be added. That polylinkers may work as the multiple cloning site where you can add your foreign DNA in case of your substitution vector using the same lambda wild type genome. Some examples of lambda insertional vectors. One example is lambda ZT10 and second one is Sharon 16A. In case of lambda ZT10, there is a eco-R1 site at the CI gene which can be digested and where the foreign DNA can be inserted. In case of your Sharon 16A, engineered lambda LACZ gene is there in the genome with an eco-R1 site which can be digested and which can be used as your cloning site for insertional vector. Examples of replacement vectors EMBL4 and Sharon 40 are some of the replacement vectors based on lambda genome. Here the stuffer region can be replaced with polylinkers which can be used as multiple cloning site where you can put number of fragments. Another example of lambda based vector is the M13, which bacteriophage M13 has got a uh, cycle of single-stranded DNA which can be manipulated, which is useful during sequencing and all. You can see here some genes like REP region encodes protein that are important for DNA replication. There are sequences called as CAP and MOR regions containing genes that specify functions associated with capsid formation and phage morphogenesis respectively. The vector M13 is identical except for the original orientation of polylingual region. There are M13 based vectors which can be used for purposes like DNA sequencing and all. Here some of the commercially available lambda based vectors: lambda z expression vectors, p blue scripts, supercos, etc. Lambda EMBL. These are lambda EMBL34 is a general vector, not a expression vector. You cannot get a product of your clone gene, while lambda zap and p blue scripts are expression vectors where you can get your product. It will be another class of vectors is cosmid vectors. You know, cos sites are required for packaging of bacteriophage DNA into viral particles. Now, this cos site is engineered into a plasmid vector to generate cosmids. The major advantage of cosmids are the carrying capacity. It can carry up to 47 kbs of foreign DNA. The recombinant cosmid DNA is injected into a bacteria and circularized like fast DNA and replicates as a normal plasmid without the expression of any fast function. Transform cells are selected on the basis of resistant gene markers. This is the normal strategy of cloning using cosmid vectors. The basic features, if we can see, there will be origin of replication, antibiotic resistant genes, cos sites, and some restriction sites where you can add your foreign DNA, which is the simplest form of cosmid vectors. But when it becomes an as expression vectors, you have to add some more sequence. This is the cloning strategy. We have obtained this diagram from standard textbooks. We have already mentioned in your curriculum. You can see here digesting the cos site gives you cohesive ends and which can be ligated with foreign DNA packaged into bacteriophage and which can again be infected into host bacterial cells. High capacity cloning vectors, some of the examples are PAC, BAC and YAC. PAC carrying capacity is around 100 kbps. BAC is based on P1 bacteriophage. It contains certain sequences which is unique packaging site called as the PAC, which help in packaging in vitro. Contains LOXP, which actually recognized by the phage recombinase, the product of the phage CRE gene, which leads to circularization of packaged DNA after it has been injected into the E. coli host cells, expressing the recombinase. Clones are maintained in E. coli as low copy number plasmids. High copy number plasmids can be induced. P1 system has been used to construct genomic libraries of mouse, human, yeast and Drosophila. Some of the successful examples. Cloning strategy using P1 cloning vectors. Back again has a higher amount of carrying capacity up to 300 kbps. Earliest back system was based on single copy sex factor F of E. coli, the simplest ones. The back may contain lambda, cos and n sites which are from lambda files. locks P site from P1 bacteriophage, also contains multiple cloning sites, contains GC reads restriction sites, T7 and sp6 based promoters in case of your expression vectors. In commonly used back multiple cloning site contain reporters in LACZ for screening transformants. There is sequences in a back normal back vector. You can see REP, E or ES, kanamycin, PAR A, PAR B, etc. Yeast artificial chromosome. This is a linear kind of vector. It contains telomeric sequences noted as TEL, centromeric sequence CEN, and autonomously replicating sequence ARS, which are important for YAC vectors. YAC selectable markers usually do not confer resistance to antibiotic substances. That is important, as in E. coli plasmid, but instead enable growth of yeast on selective media lacking specific nutrients. That is the selection criteria for yeast artificial chromosome. Your media will lack something and the transformant will survive in that media through the genes present in the YAC. YAC sequences contain ARS1 yeast sequence of replication, CEN4 telomeric sequence, TEL telomeric sequence. CEN4 is actually centromeric sequence, TRP1 selectable marker, URA3 selectable marker, SUB4 has a reporter gene, and which also can be a circularized vector. Vector insert sizes and their host and capacity. In some cases, you might be requiring this information. Expression vectors. Expression vectors are required if one wants to prepare RNA probes from the clone genes or to purify large amount of clone gene products like RNA and protein. Usually a promoter specific to the vector is used. Can be regulated easily by changes in growth conditions of the host cells. That is important. Expression vectors should contain some sequences like promoters, polyadenylation site, transcription termination site, and ribosome binding sites. Besides, there will be MCS, selectable marker, origin of replication, etc., which we can see here. It's a generalized mammalian expression vector. We can see here MCS, multiple cloning site, selectable marker denoted as SMG, eukaryotic promoter denoted as P, polyadenylation site PA, and termination site TT, and intron I enhances the production of heterologous proteins. That sequence is important if you want to get a protein product of the gene. Propagation of vector in E. coli and mammalian cells depends on origin of replication. There are 3-4 different origin of replications possible. So it's a binary vector. You can express it both in E. coli as well as in eukaryotic cells. So two origin of replications are there. Besides, there are ampicillin resistant genes. In some cases, depending upon the purpose, we may add more sequences, such as secretory proteins. If you want to express, we need some sequences called as your linker sequences. signal sequences, sorry, required for secretion, protein affinity like T, proteolytic cleavage site P, and stop codons like SC. And besides, there will be 5' UTRs and 3' UTRs regions which are important for eukaryotic gene expressions, which we have already discussed in our molecular biology classes. So here, if you want to express a secretory protein of eukaryotic origin, we require Kozak sequence, AUG start codon, signal sequence which will lead the protein to its destination, secretion protein as protein affinity tag T, proteolytic cleavage site P, stop codon SC, and UTRs 5' and 3' UTRs. This is a very good example of expression vectors expressing a eukaryotic protein in host cells for secretion, which will be secreted out of the cell. Promoters. What are promoters actually? It is feasible to modulate the expression of defined genes in an organism by combining them with a promoter of choice. That means if you want to get a product of your clone gene, the first important thing you should have in your vector, in your construct, is the promoter. Promoters have huge influence in biotechnology, especially in GMOs. All transgenics contain promoters of choice. That's why we also have a branch of study called as promoter engineering to express more and more product of a particular gene in a host system. We can engineer the sequences of the promoter region by which we will get more of the more product from the sequence. Promoter sequences along with some other sequences can be added to a cloning vector to produce expression vectors, which we have already discussed. Now there are three types of promoters. One is constitutive promoters. They direct expression of genes in virtually all cells. There is nothing like tissue specific expression. They control expression of especially the housekeeping genes. Examples: cauliflower mosaic virus and 35S promoters, which are usually used for transient expression in plant tissue culture. Tissue specific promoters is the second group of promoters. These promoters direct the expression of a gene in a specific tissue. For tissue specific expression, we need tissue specific expression promoters. For example, you have cloned a gene into a plant and you want to express particularly that gene in root cells of the plant. Then you'll have to use a specific promoter which will promote expression in the root cells of the plant only. Inducible promoters. They are highly interested in case of genetic engineering as their performance depends on certain endogenous and external factors. That means you can control their expression from outside by putting some chemicals or some stress or some creating some environment. Some possible vectors for plants and animals. You can see here Some of the examples like PI plasmids of agrobacterium fumigations, which is very important for plant transformation. Again, called for mosaic virus, Gemini virus. These are extensively used. In case of animals, there are reporter genes which are more important. We will be discussing in your third part of our presentation. Selectable markers and reporter genes, there is a difference, and some of the very good examples of selectable markers used both in prokaryotes and eukaryotes are mentioned here. Now for cloning blunt-ended DNA into a vector, we need to do some modifications. One type of modification is the use of linkers and adapters. Linkers are chemically synthesized double-stranded DNA oligonucleotides containing one or more restriction sites for cleavage by restriction endonucleases. When we want to clone that fragment, we can digest it with restriction enzymes. We will get overhangs and we will be able to ligate it into a vector using ligase. Linkers and adapters, there are differences. Linkers are double-stranded DNA which are cut into by restriction enzymes, get overhangs. Here, the cloning strategy involving linkers is shown. See, you can see linkers are double-stranded DNA which is cut using restriction enzyme. Then we get a overhang. That overhang actually helps in cloning the foreign DNA into the cloning. Similarly, in case of your adapters, you have the overhangs present already. So you can directly go for ligating the fragment into the cloning vector to make your